All right, the next few videos we're going to be talking about genetics, the branch of biology that sort of deals with probabilities and inheritance and the way in which different alleles of different genes interact. And in this particular video, we're going to be focusing on what's called the law of segregation, which, as you are going to see, is nothing more than just the consequences of some of the activities that occur during meiosis. Now, let's remind ourselves of what homologous chromosomes are. And I reminded you when we were talking about meiosis that this was going to come back and this is going to be extremely important. So if we have a diploid organism, what that means is for every chromosome, that individual has another one like it. So for example, if we look at this red chromosome here, since this organism is diploid, there's another one that looks like it. This one here, this blue one. Now these aren't sister chromatids. These are homologous chromosomes. That means that although they are similar, although they have the same shape, although the centromere is in the same location, although they have the same loci, meaning the same types of genes in the same location, the actual version of those genes, the allele of those genes can be different. What do I mean by that? Well, we have the gene for flower color here, and we have the gene for flower color here in the exact same place on the homologous chromosome. The difference is that this is the purple flowered allele of the flower color gene, and this is the white flower allele of the flower color gene. They have, right, their sequence is quite similar, right? TAAATC, right? TAA, but there is one difference. Okay, now those differences between alleles might be quite dramatic, or the differences like we see here between two alleles, the differences in sequence could be quite small. Here, for example, the difference between the two alleles, between the white flower allele and the purple flower allele, is one single base difference. And that difference can be enough to completely change the trait. Now, don't get this confused with sister chromatids. So now if we look at this, this is before the S phase of interphase. This is after the S phase of interphase. This is after DNA replication. So that now we have our two homologous chromosomes, right? Here's our homologous chromosome, here's the other homologous chromosome, but each one of them are now duplicated. So each one of those homologous chromosomes consists of sister chromatids, which means that they are 100% identical to each other. So if we have the purple allele of the flower color gene here, then it's got to be the purple allele here. If we have the white allele here, then the sister chromatid has to be the white allele. Okay, so these two guys are sister chromatids. These two are homologous to one another. So please, again, make sure you understand that, because things will become increasingly complicated if you don't get that little detail. All right, now, genes correspond to characters. So the gene for flower color gives rise to flower color, right? So you've got the flower color gene gives rise to flower color. Flower color gene is the gene, and the character that it controls is flower color. Then there are going to be alleles of those genes, various versions of those genes, which are going to give rise to the various traits of that character. So the flower color gene controls flower color, okay, which is the character. And then the various alleles of flower color will control the traits. So the yellow, or let's talk the purple flower color gene, or flower color allele, gives rise to the purple trait. The white flower color allele gives rise to the white trait. Let's take a look at a different example. So the gene that determines the seed color character. So we'll call the gene that gives rise to the seed color character Y, WHY. And then we have two alleles. We have the capital Y allele that gives rise to the yellow trait. We have the little y allele that gives rise to the green trait. Now, remember, we are diploid organisms. And in this particular example, in the pea plants are also diploid organisms. And so that means that every organism has two copies of a particular gene. So every organism has two, quote, WHY genes. If both alleles of that gene are identical, so capital Y or little y, we call them homozygous or true breeding. Okay, so in this particular case, we have homozygous here and we have homozygous here. And in that particular case, it's really easy to predict what the trait will be. If we have homozygous big Y, we're going to have yellow. If we're homozygous little y, we're going to have green, because the little y gives rise to the green trait and the big Y gives rise to the yellow trait. Now, the complicated part is, well, what happens if in our diploid organism one allele is the two alleles are different? So in this particular case, this is a heterozygous state where the organism has two different alleles for a particular gene. In this case, our Y gene, we have a capital Y and we have a little y. So what we do, the only way to figure out what that trait is going to be is to actually set up this particular, what we call genotype. Our heterozygous genotype. We set up a cross such that we cross our homozygous big Y to our homozygous little y. We cross those two together and we see what we get. So we know 100% of the offspring must be big Y, little y, and we look, what is the trait that they display? If that display is of one of the two parents, so in this case, the big Y parent, then we call big Y dominant. And the little y then is recessive, because when you put them together, big Y and little y, all you see is the trait that corresponds to the big Y, yellow. Okay, now this big Y, little y is a heterozygous state. We have two different alleles at the seed color gene. And this is no longer true breeding. If you cross this to itself, you're gonna start to see some green seeds come out. All right, so lots of words and vocabulary here, and it's really important that you understand that. Again, how do we determine what's dominant and what's recessive? Well, you take a true breeding of one allele, you take true breeding of the other allele, you cross them together, and whatever trait you see, the allele that gives rise to it, that's the dominant allele. Very important to understand. Now, oftentimes we're always talking about letters, big Y, little y, big Y, little y. But remember, these letters correspond to genes that have DNA sequences that lie on chromosomes. And if they are the alleles of the same type of gene, they must lie on homologous chromosomes. So here we have the big Y lying on this homolog. We've got the little y gene on this homolog here. Okay? The big Y and the little y are both alleles of the, quote, Y gene, which controls the seed color character. All right. Now, let's talk about the law of segregation. And the law of segregation says that when you make gametes, when you make sperm, when you make eggs, because they are only one set of chromosomes, because they are not made up of homologous chromosomes anymore, each gamete is only going to have one allele of each gene. Each gamete is either going to have a big Y or a little y. It cannot have both because sperm and eggs, so gametes, are monoploidy or haploid. They do not consist of homologous chromosomes. And that is just, right, as a result of anaphase 1 of meiosis. Right? That is the reason for the law of segregation. That is segregation. So let's remind ourselves. If we have homologous chromosomes, they will eventually, if we're going to go through meiosis, they're going to eventually duplicate. So now we're made up of sister chromatids. And then remember, in metaphase 1 of meiosis, they're going to pair. The homologous chromosomes are going to pair along the metaphase plate. And then during anaphase 1 of meiosis, those homologous chromosomes go to opposite cells, opposite sides. Basically telling us that any given gamete is only going to have one allele of each gene. It's not ever going to have a big Y and a little y. It'll only have a big Y or a little y. So when we're sort of practicing in Mendelian nomenclature, when we're not kind of looking at chromosomes anymore, we're just sort of looking at letters that represent chromosomes like this. When we look at, okay, here's a big Y, here's a little y, now we're going to take this through meiosis to make gametes, to make eggs and sperm. Well, it's going to end up with a sperm or an egg with a single big Y, or a sperm and an egg with a single little y. That's it. Those are the two types of sperm that can be made, or the two types of eggs that can be made. Why? Because of the anaphase 1 of meiosis. So, if we want to start to put together some math and figure out what is all the possible offspring that can come about from a cross between two heterozygous individuals, this is referred to as a monohybrid cross. Mono because we're talking about a single gene. The gene that controls the seed color character. Hybrid because both individuals involved are heterozygous. Big Y, little y, big Y, little y. It's a monohybrid cross. So how do we do this? Well, first we have to determine, let's say this is the male on the left. We have to determine all the possible sperm this male can make. Well, a big Y and a little y sperm. Then number 2. We have to determine all the possible eggs that the female can make. Well, she can make a big Y egg and she can make a little y egg. That's it. Then we are going to fertilize each of the eggs that are possible with each of the sperm that are possible and write down all of the genotypes of the offspring. We then can interpret what the phenotypes of those offspring are going to be based on their genotype. And then we can determine the probability of each of those phenotypes. And all of this can be organized using a Punnett square. Let's take a look at that. So here is dad. Dad can make a big Y and a little y sperm. Here's mom. Mom could make a big Y and a little y sperm. She can make 2 different eggs, not sperm. She can make 2 different eggs, which means the big Y represents 1/2 of the possibilities. So the probability that mom will make a big Y egg is going to be 1/2. The probability that dad will make a little y sperm is 1 out of 2, 1/2. Let's organize them now onto a Punnett square. We make a box, boop, 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 and we put here the different, on the left side here, we put the different genotypes of eggs that mom can make. A big Y egg and a little y egg. And then up at the top, we put the different genotypes of sperm that dad can make. Dad can make a big Y sperm or pollen and a little y sperm or pollen. And then we just go through and we say, okay, if this sperm fertilizes this egg, that's a Y, big Y, big Y. If this sperm fertilizes this egg, it's a big Y, little y. If this sperm fertilizes this egg, it's a big Y, little y. If this sperm fertilizes this egg, it's a little y, little y. So there are all the possible genotypes that can come about from this particular cross. Notice there's two different ways that a heterozygote can get made. One in which mom donates the big Y, one in which dad donates the big Y. Now we can overlay the phenotypes over these genotypes. Since we know that big Y is dominant, that yellow is dominant, we know we have a yellow here, we have a yellow here, and we have a yellow there. And since we know that little y is recessive, we're only going to see it when it is in its homozygous state, and so only this is going to have green seeds. So our phenotypic ratio here is going to be 1, 2, 3 yellow to 1 green. 3 dominant to 1 recessive. And that is a sort of ratio that you will see anytime you're looking at a monohybrid cross dealing with alleles that have a dominant recessive relationship. Okay? Now, if we were to ask a question in a different way, basically, what is the probability of generating an offspring that is yellow? Well, the way you figure that, the way you answer probabilities is you always answer them in fractions, not ratios. So fractions means that the denominator is going to be the total possibilities. 1, 2, 3, 4. Okay, there's 4 different possibilities that one can have. Now, the numerator is going to be what fits your search criteria. You're searching for the dominant phenotype, yellow, so that is 1, 2, 3. So the answer to what is the probability of generating a yellow offspring with this cross is 3 out of 4, or 3/4. Please, when you're answering these questions, do not do percentages. Please do not do decimals, right? Make sure you keep them in the fraction form. So here is a list of the important terminology here. That you're going to have a gene, and the gene gives rise to a particular character, like seed color. Then of that gene, you're going to have lots of different versions, lots of different alleles. They are going to give rise to the various traits of that character. So within the seed color character, we have yellow seeds, we have green seeds. The genotype is the alleles that you have, the big Y, the little y, the little y. The phenotype is the actual traits that you see, the yellow or green or white flowers or purple flowers. A homozygous state is going to be when the 2 alleles of a diploid organism are the same. Heterozygous state is when they are going to be different. In the next video, we're going to talk next in regards to the law of independent assortment.